In this video, we will be discussing point mutations and how changes in genetic information alter the finished product of transcription and translation. This is the sequence of DNA we will be using for our discussion. Let's start by finding the normal protein product, which in this case is 5 amino acids in length. We must first establish the reading frame by finding the part of the sequence which will transcribe to the start codon. We can do so by looking for the letters ATG in the DNA. When we find it, we know this is a coding strand and the other is the template strand. We will then read the DNA template strand from 3' to 5' and transcribe it into mRNA until we find a stop codon, UAA, UAG, or UGA. Now we will go into translation. Beginning at the start codon, AUG, we will read it in frames that are 3 nucleotides long and find the corresponding amino acid on our amino acid chart. In the 5' to 3' direction, we have AUG, which codes for methionine, GUC, which codes for valine, AGG, which codes for arginine, UUC, which codes for phenylalanine, AAG, which codes for lysine, and finally UAA, which is our stop codon. Now we can get into the mutations. A mutation is just a change in DNA. We will be dealing more specifically with point mutations, which are substitutions of one base for another. These will either be transitions or transversions. A transition is when the change is from a purine to a purine or a pyrimidine to a pyrimidine, while transversions are changes from a purine to a pyrimidine or a pyrimidine to a purine. These mutations have 4 different effects on the end product: missense, nonsense, read-through, and silent. Missense is when the point mutation causes an amino acid to be changed to a different one. Nonsense is where a normal codon changes to a stop codon. Read-through is the changing of a stop codon to an amino acid, so translation continues to occur. And silent is when the mutation has no effect on the final product because the mutation coded for the same amino acid. Here's an example of a missense mutation. In the DNA coding strand, a G is changed to an A. When transcribed, the third codon no longer codes for arginine like it initially did, and now codes for lysine. This means that the protein product is different and may not be suitable for its intended function. Since G and A are both purines, this is a transition. Next is an example of a nonsense mutation. In this one, an A is changed to a T in the coding strand. When transcribed, this T becomes a U, changing the codon for lysine to a stop codon. This ends translation prematurely, and the protein product is only 4 amino acids in length instead of 5. This is a transversion because adenine, a purine, is changed to thymine, a pyrimidine. The third is a read-through mutation. This time, the last nucleotide in our established reading frame mutates from an A to a C. When transcribed, it reads UAC instead of UAA. UAC is no longer a stop codon and codes for tyrosine. This means that the translation of the mRNA would continue and would be longer than it is supposed to be. This mutation is a transversion, since A, a purine, is changed to C, a pyrimidine. Finally, we have a silent mutation. In this case, a C in our coding strand changes to a T. When transcribed, we get UUU in the fourth codon instead of UUC. Luckily, both UUU and UUC code for phenylalanine, so the end product of translation is the same and the function of the protein is not affected.